I'd like to thank the organizers for the opportunity to present our work on atopic dermatitis and the use of our non-invasive platform in assessing skin inflammation. First of all, I'd like to give a little bit of a background on the disease itself. Atopic dermatitis is one of the most common chronic inflammatory skin diseases. It primarily affects up to 25% of all children and 10% of all adults. In addition to the skin disease, it's associated with a significantly impaired quality of life due to the vicious cycle of intense itching, scratching, and disruption of sleep. In addition to the impact it has on the quality of life, further characterization of the lesional skin has identified an impaired protective skin barrier, so the skin barrier function, for example, deficient innate immune response that prevents its ability to defend itself against invading bacteria, and is associated with increased T-cell mediated inflammation. Specifically, atopic dermatitis lesions are predominantly characterized by the increased expression of Th2 cytokines, Th22 cytokines, and epithelial cytokines. As you can see on the right side of your screen, this is very multifactorial in terms of the nature for atopic dermatitis. There's a lot of overlapping inflammatory mediators and factors that play a role in the regulation and pathogenesis of this disease. This makes it very difficult when thinking about how does one actually identify those populations and who's going to benefit from a particular therapeutic. Specifically, about 60% of moderate to severe atopic dermatitis patients do not achieve clear or almost clear with current biologics that target the Th2 axis. So at DermTech, we use a non-invasive platform to assess skin inflammation. On the left side of the screen, you can see the Smart Sticker. Our Smart Sticker is a 19mm adhesive patch that's used to collect the outer layers of the skin. The image on the left of your screen is a Smart Sticker that's been used to collect the outer layers, and you can actually see the skin cells attached to the specific Smart Sticker. What this allows us to do by using a non-invasive platform is that with 4 total tapes, we can actually collect 1.5 milligrams of skin tissue from the outer layers of the epidermis. This is a representative skin sample of the entire full thickness epidermis. How we're able to do this is that the basal keratinocytes, as they go through the terminal differentiation process and migrate from the basal layers to the outer layers or to the stratum corneum, while they do become a nucleate, they still retain the protein, RNA, and DNA materials that were formed with. This allows us to go through and non-invasively capture the outer layers of skin without disrupting the lower levels of the epidermis. Specifically, in this study, we analyzed 31 subjects with moderate to severe atopic dermatitis and 20 subjects with healthy-looking skin. The skin samples were non-invasively collected using the DermTech Adhesive Skin Collection Kit, and each kit contains 4 Smart Stickers that allow us to collect the skin cells as deep as the stratum granulosum, and then preserve those skin cells for RNA extraction. Due to the proprietary nature of the Smart Sticker, the material, the genomic material in this case, is preserved for up to 10 days at room temperature, or if we need it for longer storage, we can store it at -70 for over a year. Following the RNA extraction, inflammatory genes were assessed by real-time PCR. In this instance, we specifically focused on a targeted set of genes to understand the basic heterogeneity and the differences between normal-looking or healthy-looking skin and lesional atopic dermatitis skin. On the upper left of your screen, you can see CCL17 and TARC. This is a known biomarker that correlates with disease severity in atopic dermatitis patients. As you can see, there's a significant increase between the normal skin and the lesional AD skin. What I'd like to point out is that each of the y-axes on all of these graphs is in a logarithmic form. So a simple 1 tick increase is actually a 10-fold increase compared to the other. So again, referencing CCL17 TARC, the levels of CCL17 TARC in the lesional AD skin is 10-fold greater than in the normal skin. When you look at the other Th2 targeting mechanisms, you can see that in IL-13, there's a relatively wide distribution of samples, where in some cases, some of the individuals have low levels of IL-13 and others have significantly higher levels of IL-13 when compared back to the normal skin. Interestingly, Our observations confirmed that IL-4 receptor alpha and IL-13 receptor alpha 1 are both similarly expressed in normal skin and in lesional AD skin. This has been confirmed by other investigators previously, and we've also seen the same thing with other previous publications looking at histopathology as well. Moving on to the Th17 axis, with a primary focus on IL-22, a molecule that's known to induce acanthosis or epidermal thickening. You can see a stratification, so there still is a significant difference between the normal skin and lesional atopic dermatitis skin. However, there's a natural distribution of IL-22, indicating that there are various levels that might be associated with baseline inflammation in these individuals. Again, IL-23, which is known to induce IL-17 production by Th17 cells, there's a significant elevation in IL-23 when compared back to normal-looking skin. Most interestingly, when we look at the epidermal cytokines, and both of these have previously been targeted by monoclonal antibodies, and you look at the expression levels of TSLP, there's relatively similar expression of normal skin and lesional AD skin in terms of the expression for TSLP. Similarly for IL-31, while there is a distribution amongst lesional AD, there are very few samples that are significantly elevated in comparison to normal skin. In addition to our normal analysis where we compared between the healthy-looking skin and the atopic lesional skin, we wanted to understand whether or not there were differences in which genes were expressed within what individuals. Again, this balanced our information off of their previous smart sticker collections and allowed us to go through and look specifically for the expression of a particular gene within an individual. If you look at the left orange bar, you'll see that CCL17, again, as a reminder, it is a biomarker of skin severity. In moderate to severe patients, was expressed in more than 95% of the patients, so 30 out of the 31 patients. IL-4 is a known transient cytokine, which is incredibly difficult to measure by real-time PCR. And again, we've confirmed the previous observations with very low levels of individuals that express IL-4. IL-4 receptor alpha and IL-13 receptor alpha 1 are expressed in a majority of cells throughout the human body. So it was not surprising to see that those two genes were expressed in 100% of the patients. Most interestingly for our observations was looking at the levels of IL-13, IL-22, and IL-23. IL-13 was expressed in 16 out of the 31 patients, so roughly about 55% of them. IL-22 was also expressed in about 50% of the population. IL-23 was expressed in about 55% of the population as well. And then when we migrate to the epidermal cytokines, we look at IL-31 and TSLP, which were both expressed in about 30 or 25% of the population. While it does confirm that there is increased expression, or at least detectable expression of those cytokines in the skin of those patients, it doesn't necessarily mention whether or not there's an overlapping signal. So therefore, we performed a correlation analysis on the right side of your screen to look at whether or not certain gene signatures or genes themselves correlated with an expression of another gene. So for example, if you look at the top row, CCL17 was obviously found to correlate with itself. And when you start looking across the rest of the row, the highest degree of correlation for CCL17 was actually with IL-23 at a Spearman correlation of 0.44. When you look through the totality of the correlation matrix, the highest degree of correlation was directly between IL-22 and IL-13, where the Spearman correlation was 0.75. Followed behind that was IL-31 with IL-22, and also IL-31 with IL-13. This suggests that there's overlapping signals are minimal, and that predominantly we start seeing an isolated induction or elevated expression of particular cytokines separate from one another, indicating that various approaches could be used in identifying subtypes of atopic dermatitis inflammation. In conclusion, overall, we demonstrate that the non-invasive skin sampling with the SMART sticker is a viable method to assess inflammatory signals in lesional atopic dermatitis skin. Additionally, we demonstrate that non-invasively assessed inflammatory gene expression has the ability to stratify atopic dermatitis patients into different subtypes. Specifically, IL-13 is known to signal through a combination of the IL-4 receptor alpha and IL-13 receptor alpha-1 heterodimer. What it does demonstrate to us is that there's a strong correlation between the elevation of IL-13 as the dominant signaling cytokine through that receptor and specific disease response to particular Th2 targeting agents. Finally, additional studies are needed to evaluate the relationship between baseline inflammation and clinical responses to targeted therapeutics. Specifically, targeted therapies that focus on antagonizing the Th2-mediated inflammation are affected in approximately 50% of the AD patients, indicating that there's a significant portion of the population that does not have a therapeutic benefit when treated with those. At DermTech, we specifically have initiated a clinical trial on clinicaltrials.gov to prospectively investigate non-invasive biomarkers in moderate to severe AD patients. Thank you again for the opportunity to present our data. Please reach out if you have any questions.